welcome back. We are talking about one of my favorite topics, and that is communities, plant communities, and the differences between sort of the closed community concepts that are easy to conceptualize if you're walking around areas that have closed concepts. That may be a Freudian slip. Closed communities, and people that hone in on that get stuck in their thinking and they have closed mind. No, sorry. Closed communities, and so then you kind of have that Clementsian viewpoint that communities are all together and that species all interact and help each other. And it's a balance of nature model. What's the huge version of that? Gaia, right? All the plants and animals are helping each other survive on this planet. But we're shifting over into the Glissonian version, which is the individualistic model, where all those species are there by coincidence, and you as a human walk into the forest and you make up all sorts of great just so stories to explain why it's this way. Maybe not full of strong data collection and hypothesis testing, because it tends to fall apart. Okay, enough of that preparation and soapbox. Let's look at some species in the eastern forests and how they seem to be demonstrating independence. So this is a figure out of your textbook, figure 18.8, from the Great Smoky Mountains. And on the left is sort of elevation on the y-axis and moisture on the x. And what you can see is as you go up in elevation, you go from red oak, hickory, through hemlock, chestnut oak, pine, up to beech and white oak. Okay. At the same time, as you go across the moisture gradient from wet to dry, you go from, beech and hemlock in the wetter areas out to pine and white oak, chestnut in the drier areas. And you can see in these little sort of heat maps, here's red oak. It can occur up and down this elevation gradient, right? But it really prefers to be up in this sort of drier, high elevation zone in competition with beech. Now the beech prefers maybe the wetter portion versus the drier, but it doesn't do really well up and down. I mean, it can go down in the lower elevation, competing with hemlock if it's moist enough. Okay, and then the white oak, which is a congener of this red oak, tends to shift towards the drier habitats, staying away from the red oak potentially. I don't know about staying away. There's a human interpretation, but they do tend to separate. Then there's even a disjunct population, not population, but habitat preference that possibly occurs in this sort of medium moisture gradient, low elevation. So looking at that, my question there on the left, where would you go to find all three species in one community? Can you? Looks like right here at dry, high elevation, you could find red oak. If you look carefully, you could find beech. If you look carefully, maybe it's hard to find the white oak there. This is the red oak. I don't know if I said white oak, but white oak over here on the far right in the drier habitats. They prefer it even drier, but they can get their spot. They can get their positions there when it's a little bit wetter. Now, if you walk into this spot right here that I just highlighted and you see all three species and you're Clements or his students, what are you going to say? Oh, there's an association. They all need each other. If you're me, you look at that and say, wow, coincidentally, they're all here at the same time. And I'm here too. So I get to observe it, record it, and figure out why. But it may not have to do with the different species being there at the same time. Have I made my point yet? So here's another example of that same idea. This out of your textbook, or maybe it's an earlier textbook since I don't seem to have the figure number here. But it shows you the range of a variety of different plants or trees and how they converge in Kentucky, a lot of them. So you can get this really diverse forest in Kentucky. Some of the species prefer it north, some prefer it south, some prefer east, some prefer west, upper elevation, low elevation, moist, dry. It just happens that in Kentucky, the conditions are pretty good for a bunch of species. And they all converge here. Now, you walk into that forest and you say, oh, okay, here is a community, and Clements is telling me that this is a superorganism. They all need each other. Okay, but this is a snapshot in time. This is one researcher maybe who has gone around and pulled out all the data to figure out what are the ranges of the different tree species and mapped them out and sees that something's going on there in Kentucky where they're all together. But it's the life of a human in terms of successional changes and changes in ranges, etc. So what's going on today may not have been going on 10,000 years ago or 18,000 years ago. So here is some data some data from pollen records in ponds looking at what tree species were present on the landscape from Florida up into Canada from 18,000 years ago up to about 5,000 years ago. Okay? No, 500 years ago, excuse me. And what you can see is, let's just look at ironwood. Nothing, 18,000, no pollen in any ponds in our eastern forests 18,000 years ago. It starts showing up in pockets 14,000 years ago. Now the gray area is the continental ice sheet, and it's melting 18,000 years ago. It's starting to retreat north. And maybe with that climate change pattern, that allows ironwood to get established, to do quite well by 12,000 years ago. And then it's still moving north, but it doesn't seem to be holding its footing. Meanwhile, the hemlock, starting in Florida, moving north, starting another batch as time goes on, and that's probably moving north also. So there seems to be two different rounds of hemlock establishment. Oak, south, moving north, and basically colonizing the entire continent as the ice sheet retreats. Spruce is more likely, or seems to be more likely to be moving with the ice sheet north with climate change. Okay, so that Kentucky picture doesn't take into account the fact that all these trees have their own migration pattern, post-glacial migration pattern. And so we, again, with our human sense of scale, can make some very incorrect assumptions about associations and what trees need what trees around. Strongly coincidental, I would argue. All right, now that last point should raise in your mind the idea that scale is really important. And I don't mean like the insect pathogen on trees or your houseplants, nor do I mean a guitar scale or piano or something. What I'm talking about is the scale of the observation. What's your temporal scale that you're thinking about when you see this pattern and make up your story based on the data you're collecting? Are you only honing in on the last, 5 to 10 years that you've been walking through that forest for your master's or your PhD or your undergraduate research project? Or are you allowing for conditions to be changing constantly and that there's a much longer time scale involved with the trees that are there, a much greater spatial area that they are interacting with each other under? And can you get better images with those ideas? So I do want to emphasize that an important defining feature, remember this section was all about how do you define plant communities or communities, is the scale in space and time, temporal and spatial. And they're usually linked, those two ideas. So big spaces, longer times. Small spaces, shorter time scales. And I give you an example is a log that is a community at one scale. Now I pulled this frog out of the textbook. I don't think it's one that lives in logs. I think it prefers creeks. But we could just say, okay, instead of a log, we're talking about a creek. And its one scale, it's the world for that frog. But at another scale, it is basically something to step into and move on for a deer, right? Or even a larger frog that basically has a larger spatial scale that it interacts in the landscape on. The temporal processes may be very different for that log or creek. We'll stick with the log here. It's probably occurring within a few years. That log is gone. It's decomposed. It's gone from a log that fell to basically a humus layer in the soil, turning it brown. So through decomposition from insects and hikers stepping on them and their boot crashing through, obviously fungi, white rotting fungi and brown rotting fungi, and bacteria and all sorts of things. There's a lot going on in that log for a few years and then it's done. The forest that that log came out of will be there for hundreds, potentially thousands of years, you hope. Climate change impacts that forest, not so much the log, right? Maybe with warming temperatures, the rates of processes are increasing. It's getting faster, it's decomposing quicker, but not the way the climate is altering where those trees can get established. And the forest has a much more kind of coincidental time scale with climate change than a dead log that has a microbial community in it. And that's largely because of the process rates are different. That log, the process rates are on the orders of 24 hours, 12 hours, maybe a week, maybe a year, whereas the forest process rates are much longer, right? Reproduction, seed falling, most of them dying. One starts the next generation over the course of the life of the tree. So as you can see, the process rates are very, very different. All right. Where am I? He says to himself. Okay, there I am. So we do need to define our communities if we're going to understand what's going on and be able to do experiments that are meaningful and tied to that community. But the best way to do that at this point is temporal and spatial. However, that's not particularly helpful with a lot of the biotic interactions. So one of the ways we define communities a lot, especially at this university, is through our understanding of diversity. So what species are there? How many species are there? What are the assemblages? And we can kind of build out the ecological stories from there rather than the temporal spatial area that's involved. But the temporal spatial area is important for understanding the system. We tend to be more interested in the biotic components. And so measuring species diversity is a huge part of ecology. And again, especially at our university. So let's look at some of that. We're going to start with a very simple version called species richness. Species richness is just the raw count of species in the system. So if we go through the cemetery near our campus at ESF and start recording the number of birds that are there, we'll come up with a concept of how many species are there. If we just go out once a week and sit under those trees in one location and record all the songs, if we're good about that, or visual identifications, we're going to have a certain number of species at the end of 3 weeks. Or if we extend it out 6 weeks, we might get additional species, maybe even because of migrations, but we have to take that into account also. And so it's just the raw number. And so you're going to very quickly record the presence of a chickadee, right? You might even see or hear a pileated woodpecker, okay? Very different abundances, but it doesn't matter with species richness. It's just, have you seen it? Yes. Presence, absence. And it doesn't matter if you see 400 or 2. It has the same weight in your count for species richness. So it's the number. It's a relatively easy way to quantify diversity. But because it doesn't take into account abundance, it doesn't take into account something like crows are incredibly abundant here, and they're dropping all kinds of nitrogen into the system and on the sidewalks, and okay, you get the point. So abundances are important and a little more difficult to quantify diversity when you start taking into account abundance. But that's what we're gonna do next. And you're doing it in the lab if you haven't already, with insects, soil microbial arthropods, but also fish in our local creeks. So richness has its problems. I hinted at the idea of the problem with sample size. If you go to the cemetery for a couple hours once a week for 3 weeks, you'll get a list of species. But if you went for 6 weeks or a year every week, obviously you're going to get additional information. You're going to see types that are rare that you just don't get to see just on a random set of 3 weeks. Okay? But if you had superpowers and you could look at every single bird every time you went out there, you might get a complete picture, except for the migratories coming in and out. So sample size matters. The more times you sample, the more likely you pick up the rare types. Best way to put it. And abundance matters. So you go out there once, you are going to see a crow. You are going to see a chickadee. Okay? If you go out there a whole bunch of times, you may pick up the pileated. Or even if you're extremely lucky, which I have not been yet, you might pick up a snowy owl. Okay? Okay, this is what's called a reverse J-shaped curve. I like it because it begins to show us abundance, not just species richness. So you get all your species out there and you start to quantify how many are there each time you measure. And you get a mean for the abundances of each species across your sampling scheme. And so on the x-axis we have the number of species up to 15. It's a fairly depauperate richness. But then it shows how frequent each species is across this x-axis from not very frequent at all, down 0 to 20%, up to very frequent. I shouldn't use percent there, but a frequency class of 0 to 20, up to 100. So 100, 80 to 100, those are our crows, those are our chickadees, extremely abundant, but not a lot of species that have that kind of abundance, right? Not a lot of species are so abundant. There are a lot of species that aren't very abundant at all. In any given community, we could change this axis to say 100 different fungi, or 1,000, or microbes in the soil, right? You're still, if you mapped it out, you'd still get that reverse J-shaped curve where a few aren't very abundant, but you will pick them up if you sample enough. And then there's a lot of them are in that way. Not very abundant. Most species are very infrequently encountered. But there are a few species that are very abundant. And let's see, soil microorganisms, various colembula, for instance. And then some of the colembula might be really abundant, whereas others are quite rare. So same reverse J-shaped curve across the board. And in words on the slide, most species are infrequent. And I write, think snowy owl. A few species are very frequent. Think crow, chickadee, blue jay. In my backyard, currently, goldfinch, right? Over on this side, somewhere in here are the various woodpeckers, and even the red-tailed hawk is making a strong presence this year. By the way, that one pops up because we have one of those trampolines with a netting around it. The red-tailed hawk got inside and kept bumping into the netting. So my son saw it. I walked out there. I went in. It was hanging on the netting. This was about 3 days ago, so it's still in my head. I sat down and I started to tell the thing it needed to just fly up because it's open. And eventually it let go. It was hanging upside down with its talons stuck in the netting. Then it started to fly up and I crawled over underneath it. I kid you not, I could show you the video that he's not a great videographer, but crawled over and the thing flew up, and so there's me standing up and like I'm looking at something. Go, fly, be free. Later, everybody I've shown that to said, you're a nut, because that thing could have attacked you. But it's a baby, it's been around, I've been talking to it, we have a thing. Okay. Let's see. All right, now we're going to look at how to measure abundance beyond that reverse J-shaped curve. And make sure I'm in the right place. at the right time. Yeah, I'm going to power through here. So there are two main diversity indices. There are many, and computer programs provide a lot of opportunities here for you to take your data set, the raw data, the observed frequencies and richness, or I should say abundances and richness, and then put them into a computer program, and based on your sampling, understand what the true diversity might be. So you have your observed and your expected. Another way to do this is with these indices, the Simpsons and the Shannon-Weiner. So D is the Simpsons index of diversity. It's just 1 over the sum of the proportions of each of your species squared. You're going to use that in the lab, okay? And you can look in the textbook. I will show you a slide in a minute so you can see what this means. Right now it just is mumbo jumbo, but this Greek S, the sum of all the proportions of each species that were squared. And that gives you D, okay? I'll show you how to interpret that in a minute. The Shannon-Weiner is related. It's also looking at, sometimes called the Shannon-Weaver. I'm not too sure why, I haven't learned. But it's represented by H, and it's the negative sum of the proportions times the log of the proportions. It turns out it gives you a pretty similar number. So essentially it's just squaring, right? This is squaring straight up. This is squaring by the log, or I should say multiplying by the log, but it's still proportional data in both areas. Okay, so in both equations, P sub I is the proportion of each species in the total sample. So you're going to get a single number. You've just lost all the information about what species you have in your system, but you have an understanding of how many. Same with species richness. You don't see how many species or what species are there. You just have a count of 200 or 10. You lose all that specific information. But it's meaningful, and you can compare communities, pre-fire, post-fire, changes in elevation, okay, etc. You add the other information, that is the species identities, in another portion of the paper. So here is a table out of an old version of your text, which is to say, I'm not happy that they took this out, because I think it's really helpful in our class. We tend to hone in on these kinds of indices in EFB because we really are focused in on diversity and evenness and what is the contribution of each species in the community. So I'm going to walk you through this. The slide is available obviously in the YouTube presentation, and so you can come back to this again and again, especially when you start writing up your paper that might use this kind of analysis. So we have 5 different communities that we're comparing. Community 1 through 5 on the, what I would say, left-hand side. Okay, and then you have 5 different species, A, B, C, D, and E, and in each community, the species have a different proportional contribution to the total number of individuals in the community. All right, here's Simpsons, D, and here's Shannon-Weiner. Well, actually H here, but if you take the natural log raised to the H, you get a number that is basically equivalent to what you see with D. All right, again, I'll explain this in a minute. So in the first community, let's look at it. Each species is making up 20% of the whole community. So 20% for A, 40. If we add in B, add in C, you get 60% of the community, 80% with D, and then finally with E, 100% of the community has been described with each species basically contributing 20%. A very even distribution of species in this community. How many species, if you were to write this up as species richness? 5, A through E, okay? If you calculate D, look what you get. It's an even community, so it is 5. Or E to the H, 5. So that tells you when richness is very similar to the diversity index, whichever one you use, you've got a very even distribution. And sorry for this next little cartoon. I was bored last night at 11 p.m. There's 5, shouldn't be so thick. and there's 5. So our two indices are giving us the same information as the richness. They're all very much in concert because of the evenness. Now let's drop down to community 4. Now you see that species A makes up 25%, B 25%, C 25%, C 25%. Well, that's 100% of the community, but we do occasionally see an E species, so we add that in also. This is no longer a very even distribution. Actually, it kind of is. The first 4 are very even. The last one, not so much. So our indices of diversity show that. We've gone down from 5, even though the species richness is 5, we've dropped it down to 4. If you do the calculation using those equations in the last slide, okay? So and then it's 4.02 because of the little bit of contribution species A provides. And then note that we start to see a separation between Simpson's and Shannon. Now, this one gives us a slightly higher number. And the reason for that is that this one tends to weight rare species more than Simpson's. Okay? So this is 4.02 because it weights the very frequent species more, or at least the rare species less. And this one rates the rare species more. And you can see that even more so in the last one, Community 5, where we have 50%, 30, 10, 7, and 3%. And now, again, it's less than 5. The richness is 5, but the diversity tells us 2.8, telling you that it's not a very even distribution of species or frequencies. And the other, Shannon Weiner, actually ekes up closer to 4 because it weights the rare types more, whereas the Simpsons is weighting the more common types more. So this one says 2.8 because we're starting to lose our diversity. This one says 3.42, less than the 5 species richness, but those rare types are there, and we need to account for those. So a lot of people are always asking me when they're doing some kind of analysis for their graduate work, you know, what's the difference between these two? And my pat answer is that one of them weights rare species more, so you've got to decide if that's what you're looking to address or not. Okay, and they are giving us evenness. I want you to be sure you see that. Very even tells you, if they're all the same, 5, 5, 5, it's a very even community. As the numbers start to separate, you become aware that they are less even in that community, those species. All right, that was a fairly fun slide. Couple more, 3 more slides on this topic here. So Simpsons, this kind of goes over a lot of what I just said. D can vary from 1 to S, and S is richness, the number of species in the sample. The larger the S, the greater the diversity, and the greater the evenness. Closer it is to the richness, the more even the community. Okay, and let's see, when all species are equal, D equals S. When there's unequal, D does not equal S. Could be less, probably is less, it is less. Rare species contribute less to the Simpsons than the common ones, and the same is true for the Shannon Weiner, or again Weaver, depending on which, there seems to be 2 different variations on the name here. But it's very much like Simpsons, and it gives a little more weight to the rare species than the Simpsons does. That's the main difference. But you have the equations 2 slides ago to tell you, and those of you that are mathematically inclined can look at that and say, oh yeah, it totally makes sense. When you throw in, instead of a square of the proportions, you say the proportion times the log of the proportion, that's what you're going to see, a greater weighting of rare species. All right, couple figures here using these kinds of data. This is from a different text by Moles, and he's got 2 different communities. So first, community B, very even. In fact, it's very much like our first community in the last table. So 0.2, 10% here of the population, or of the community. Up here it's 20%. This is a log scale, 20, 40, 60, 80, all the way up to 100%, 1 up here. Okay, so this is even, 20%. 20%, 20%, 20%, 5 species. Hope you're following my cursor. And they all contribute equally. Very even community. Community A, not so much. One species is almost the entire story. And then these 4 make up just a little bit. So what's that? 0.01, 0.02, 0.03, about what, 30%? I think I put 40% because if I kept counting 0.03, 4, 5, 6, 7, 8, 9, that should not be 0.1 there. The figure's actually wrong, but hopefully you can get the gist of it. These rank abundance curves for 2 different species, what is really nice is it allows you to compare 2 communities, and you could say this one's quite even. That would be community B. Pretty diverse. Community A, not so much. Looks like we have had a fire and all the fire-adapted species, which was 1, germinated, and now it's almost a monotypic stand. Just 1 plant dominating with a few that are holding on. That would be the community A down here. These not so critical in this community anymore. Okay, or something. I don't know. I'm making that story up, but not too far off. Here's where this might actually should be 3% or 4%. I don't know. Okay. And lastly, here's another variation on this theme. These are rank abundance curves. So you go in and simply measure your abundances for your communities, and you map them out on this rank abundance curve from most abundant to least abundant. So most abundant on the left, least abundant on the right. And then it's proportional abundances here again, kind of like the last one. This is a log curve. So this study was a long-term study, I believe in England, where they started fertilizing this particular field back in 1856. And they had a species richness of plants at 49. And then they're ranked in their abundance, so this is very abundant species. And then as you go out, you get to the far less abundant species up here. Okay, not very even. The slope is pretty steep. Well, not so steep, but still a little bit steep. But the point is you have a lot of species, and they each have their own abundances. 1862, we've gone from 49 richness to 28. So we're losing dots. And their abundances are changing also. All right, those that remain are becoming more and more abundant, moving to the left. Here's in 1872, only 16 left, then 10, then 8. And by 1949, little under 100 years of fertilizing, we've gone from 49 species to 3. And this was a kind of a wake-up call for, it should have been, for our overuse of fertilizers. This is probably a good piece of why our diversity is going down near agricultural settings, because that fertilizer definitely pushes the system to a less diverse system. And this is even really poor diversity because basically it's one species at close to 100% with a couple that were found. Hopefully they didn't step on them before they found them. All right, so, but what I want you to get more than just this fertilizer side story is we can compare these communities over time and see how they shift. Be very cool, and I'm sure people are doing this, to find some plots and do this kind of study as global climate change is progressing and see how that, it's not going to be an undergraduate project. We all want to get out there and solve global warming today. It's going to be a project that is started by someone and handed off to the next generation and the next generation, 20 to 30 year stretches. But keeping those data, keeping them available, passing them on to the next group. All right. So, next up then, why or is diversity so important? We'll look at that in the next segment. That'll be the last one for chapter 18. So, very good. We'll see you then. Cheers.